Hello, everyone. Thank you for coming to my presentation. My name is Vincent, and I'm a second-year PhD student from the University of Florida. I have a background in biostatistics, so this may be a bit different from what you've been seeing. And as Dr. Zelinska has said, I'm here to present my group's work on a novel gut health index. This is, the left is me. This is my co-author, Yulin Lee, and this is my advisor, Dr. Somnath Datta. I've gotten much help from him. So I'm going to be introducing the topic and data, giving an overview of our methods, and comparing our results with the existing indexes provided in the metadata. We've already had a bit of an introduction to why this is important. For a while now, we've known that there are diseases that are influenced by the gut microbiota, and since these have been increasing in prevalence and are costly to treat, it's of significant clinical interest to detect these early and be able to mitigate the costs of treatment in the population. So the stool samples can be collected frequently, non-invasively, and relatively inexpensively. And so the challenge this year has been to develop an index that can outperform the current ones that are only based on abundances using the theater of activity. And as mentioned before, the theater activity is just the statement that the microbiome should be more than just the microbiota or the species. So before we begin discussing the methods, we need to examine the data. We already got a pretty good overview from before, so I'll just go over this briefly. We have about 4,400 stool samples for over 30 cohorts and 26 unique diseases. However, while the data set contains all these diseases, we elected to bin the samples into just two groups with them being healthy and non-healthy. And healthy just means that there is its absence of disease, and non-healthy means any combination of diseases, at least one. We also did not incorporate the cohort labels into our classifiers, into our methods, because there are some cohorts who did not have one or the other, and that posed a challenge from a classification standpoint. And so our index was based completely on just the relative abundances of the metabolic pathways and taxonomical species. For our methodology, we created an ensemble index to more accurately classify healthy versus non-healthy stool samples. Our pipeline begins with feature selection, followed by hyperparameter tuning, generating the index values, and setting the threshold for healthy, non-healthy classification. For feature selection, our strategy uses a two-stage process to select the informative features. On the functional data and species data separately, we ran the Kolmogorov-Smirnov test and Cliffs delta effect size, which are both non-parametric tests and thus do not require any distributional assumptions to use. The KS tests for differences in distribution, and the logic behind us using this is that if a feature has a different distribution of values in the healthy group compared to the non-healthy group, there is a decent chance that it has a strong relationship with one's health. Now, it could be the case that the difference in distribution is statistically significant. In other words, it passes the Kolmogorov-Smirnov test, but it could be very small. So we want to filter those out, and the Cliffs delta lets us set a threshold for the differences in the distribution, so we can make sure that the features that we look at actually have a major impact, or they're likely to have a major impact on gut health. The p-value and the effect size thresholds shown here are based on values that we saw from literature for microbiome analysis. We also removed features that had near zero variance. So if the proportion of a bacteria or some species was basically the same throughout the entire 4,400 samples, then it's unlikely that it's going to have that we're going to be able to detect a meaningful effect from that with our method. So we just filtered those out, and we also filtered out uh, rare features for the same reason. So once we completed that, we were left with about 80 features total, which was a drastic reduction from the initial over 1,800 ones. And we combined the selected species and the selected pathways into a single feature matrix, uh, which was used for the hyperparameter tuning, which is our next step. So to capture a broad spectrum of learning paradigms, we selected 5 models to act as individual binary classifiers within our ensemble model. Lasso logistic regression can provide powerful results if it works well, and KNN and tree-based methods are flexible models that can incorporate interaction effects without needing to directly specify them, which is very useful to us because we don't know about all of the possible interactions between the pathways and the species themselves. So each model's hyperparameters were tuned through grid search and 5-fold cross-validation, and then saved for future use. KNN, SVM, and Lasso were tuned with the caret R package, and random forest and gradient boosted trees were tuned with their method-specific packages. So now, after feature selection and tuning, we can begin creating our ensemble index. And we're using the term ensemble because this index is calculated through the contributions of a large collection of classifiers. Now, starting with the blue box, we have the process to build this collection. First, we take a bootstrap sample of the original data with replacement. If the minority class appears in fewer than 30% of the bootstrap sample, then it is rebalanced using the synthetic minority oversampling technique, or SMOTE. Then we use this bootstrap sample to train each of the classification algorithms using the hyperparameters that we established in the previous step. Once we have the models, we can obtain performance metrics by running the classifiers on the out-of-bag data. The metrics that we used in this case were balanced accuracy, sensitivity, specificity, and area under the ROC curve. And for those who are unfamiliar, balanced accuracy is simply the average of sensitivity and specificity. Next, and once we have those metrics, we can rank the classifiers based on these metrics, and that results in a collection of ranked lists, L1 through LK. How can we select the best classifier with this list of classifiers? Through a process called weighted rank aggregation, we can create a single consensus list from the ranked lists, and then using this consensus list, we can select the top performing classifier as the member of our ensemble. Now, that is just one bootstrap sample. You can perform this many, many times. In our case, we performed it 2,000 times, and that generated a collection of 2,000 well-performing classifiers. Now that the ensemble has been created, we can use it to generate the index values for each sample. For each sample, we take the classifiers that were generated on bootstraps for which the sample in question were out of bag of, and make predictions using those classifiers and average them together. That is a bit of a mouthful, and that might be a bit confusing to follow, but just from a training and testing data perspective, it would be equivalent to only considering contributions from classifiers in which the sample was not used as part of the training data. In the rare case in which a sample is present in all of the bootstrap samples, then the index value would simply be calculated as the average of all the predictions made within the ensemble of classifiers. Once the index values are calculated, a threshold is needed so that classification can be performed. This threshold was determined by maximizing Yoden's index over the range of the threshold. Yoden's index is the sum of the sensitivity and specificity at a given threshold, tau minus 1. And we classified these samples if the index value for a sample was above this threshold, tau, it was classified as healthy. And if it was below it, then it was non-healthy. Our use of classifiers to generate the index also allows us to calculate feature importance scores, similar to how random forest does it. For a given bootstrap sample, the leave one feature out importance for a feature can be calculated through the difference in classification errors between the original data and the permuted data set, where the specified feature is permuted and others are left alone. These importances can then be averaged over the bootstraps to yield the final importance scores for each feature. After determining the threshold for the ensemble index, we wanted to compare its healthy, non-healthy classification performance to other indexes provided in the CAMDA dataset, which were GMHI, HIPCA, Shannon entropy, and Shannon entropy on functions. This meant that we needed to set thresholds for these indexes as well. According to literature, we found that GMHI values below 0 indicated dysbiosis, while those above 0 indicated a normal gut environment. The other metric thresholds were determined through Yoden's index as well, since we were not able to find specific thresholds that indicated healthy or non-healthy guts. And using Yoden's index in here kind of reflects a worst case scenario when comparing the classification performances with our ensemble. So in our comparison, we included accuracy for reference, even though it was not a metric that we used in weighted rank aggregation. And as you can see, the ensemble index performed the best in almost all classification metrics except sensitivity, but it performed comparably to the GMHI and HIPCA. In the right two columns, we also have the Mann-Whitney U-test p-value and the Cliffs delta. The Mann-Whitney U-test is another test of distribution, and these values indicate that the ensemble index has a significant and large difference in distribution of index values between the healthy and non-healthy groups. The other indexes also have a statistically significant distributional difference in the index value, but Cliffs delta shows us that that difference is not very large, and it's too small to be quantifiable. This can be better visualized in the next figure. The leftmost bar in each subplot represents the index value distribution among the healthy group, and the yellow bar that's directly next to it is the index distribution among the non-healthy group. Other bars represent the index distribution for specific diseases. There is clearly far less overlap in the healthy and non-healthy distributions for the ensemble index, which reflects its better performance as a general classifier. However, it's also undeniable that the other indexes are better able to distinguish specific diseases from each other, such as type 2 diabetes and cirrhosis in the case of GMHI, and acute diarrhea and metabolic syndrome in Shannon entropy. Shannon entropy on functions displayed the worst separation between the healthy and non-healthy groups and could not distinguish between any disease groups either. So according to our methods, we calculated the leave one feature out importance for all features retained after the feature selection, and then graphed the top 20 shown here. Several of the top scoring features were pathways, such as pathway 7237 and pathway 4903, which supports the theater of activities idea that functional data should be incorporated into indexes for better performance. The the most important one here is C. aerophaciens, which, according to literature, is linked with irritable bowel disease. Second pathway, 7237, is a biosynthesis pathway for L-lysine, L-threonine, and L-methionine. And B. pseudocannulatum has a protective effect against obesity. So there is a assortment of different like good and bad bacteria as well as the pathways. In summary, we have developed an ensemble index that incorporates both functional and taxonomical data, which is better at distinguishing between healthy and non-healthy individuals than the GMHI, HIPCA, and Shannon entropy measures. However, since this is only a binary classification, it cannot identify the exact disease or condition that someone may have. And as a result, in terms of application, this index may be used in capacity of a preliminary screening tool at the clinic that can lead to further testing if issues are found. In terms of future directions, it would be interesting if we could overcome this and enable the ensemble to predict on more than just healthy and non-healthy. In addition, there is a possibility that discarding the cohort labels could have affected performance, since there may be intra-cohort correlations that were not accounted for in our methods. In the future, when we iterate on this, it should account for the cohort labels. I would also be interested in seeing the index performance on a larger data set or a data set that's completely untrained. We did not know where to find it before, but we know now. And these are the references for the presentation. Thank you.
Thank you very much for a wonderful presentation. Have we got any questions?
Hi, very interesting. I was very glad to hear that you're going to test this on some data, because I was a bit surprised that you do feature selection before you go to do the machine learning. So you're doing the feature selection on your whole data set, and then you go and do the piece of machine learning, and that's where you do your cross-validation. So there is no complete independence of train and test, and there is a bit of data leakage there. I just wanted to flag it and see what are your thoughts.
Yes, that's a very good point. And we, my co-author and I, Yulin and I, don't have much training in biology. So the process that we went through for creating our method was to reference how other people did their feature selections. So when we looked at GMHI, we thought that and HIPCA, we saw that they looked at like good and bad bacteria, and they sort of did their own another curation for the feature selection, and we didn't really know how else to go about it. But you are right in that it's not completely independent. So we tried to have some independence in there in terms of like having out of bag, using the out of bag data for the indexes rather than using the entire sample. But it's a difficult problem.
Yeah, there's another question.
Hello. If you think about handling the labels, the disease labels, there are apparently two extreme cases when you try to predict every particular disease or just derive general healthiness index. How about exploring the middle ground? Try to cluster the conditions and identify clusters of diseases which with stronger microbiome associations.
I'm not sure I am understanding the question. Could you?
Because you mentioned you will be planning to work with more particular disease identification, and I wonder if you are talking about predicting disease down to particular condition or just clusters of similar diseases or clusters of diseases that share sort of functionality pattern.
Well, the details have not been nailed down yet, but I believe it would be easier if we were to work with clusters of diseases, like similar disease types, rather than individual disease, like specific diseases, because if we were to go with the latter route, we would be lacking a lot of data, and it's a data issue for us.
Any questions?
Hi, great presentation, by the way. I was just wondering, why did you choose the Jordan index to build the classification threshold? Is there a particular reason?
Index just gives us a balance between the sensitivity and specificity. It makes sure that like it doesn't lean too much towards one or the other.
Okay, so it was just like to keep the balance between both metrics.
Yeah.
Okay, thank you.
I have two questions. One and thank you for the very interesting talk. One very short, just to be sure I understood your positive class. It's the disease here in this, the prediction you made.
Class 1 is indicating healthy.
So okay, so the positive one is healthy. And the second question, so I would also be very interested to see the results on an independent data sets, but in the meantime, is your model, are you planning to publish or make your model available? Or?
We do want to publish a paper. It's not exactly ready yet it's still quite messy.
Thanks.
Okay, I think it's now time for the next speaker, but